Okay, shall we get going? Thanks all for coming for our last hoopla of the semester. Very nice to see you all. Jane got passed his qualifying exam, so he's smiling away in the front. We just to give you a heads up, this seminar will run on a Wednesday next semester for those of you that will be here. And wanted to thank you all for coming for this year and for this last event. Now, thinking about the CITRIS mission, remember this is CITRIS ITR. We had our very big corporate sponsors day on Tuesday, and we've got some very good vibes from our industrial sponsors. And in CITRIS, as you know, the main three thrusts now are the general service industry, which is being pushed by IBM and Microsoft. An area that we call intelligent infrastructures, which is an area that addresses things like energy, water, intelligent buildings that Professor Ahrens works on, and the general infrastructure for California. And then the third big area is healthcare. So it was a great pleasure today for me that Professor Healy was willing to speak, because this area of stem cells and regenerative modeling and so forth, you realize, if you read the newspapers, that you guys, especially the younger students here, are in an area where this is so California. You know, every other part of the country, you know, those red states and those religious states, they don't want to do this stem cell research. We understand that there's a lot of ethical issues around the stem cell research. But for those of you living and working in California, you have golden opportunities in front of you that are not there for the rest of the country, and we in California really want to try and have a mission in this area that can be globally important when other countries around the world, Korea especially, and the European countries are really pressing hard on this research area. So without further ado, I guess I'm talking a little too much and stealing some of your thunder. In the time-honored way, let's give Professor Healy a round of applause.
Thank you. Thanks, Paul. Appreciate the invitation. So I've been going around giving this lecture probably the last week or two. And I just entitled it Stem Cells, Biomimetic Polymers, and the Promise of Regenerative Medicine. And the lecture is largely conceptual in nature, to give you some maybe details of what we're trying to do from the material science side. But if you're looking for a deep sort of discussion on what materials can manipulate cell biology, I'm not going to present that today, but can certainly be happy to visit with you or sit with you and discuss some of those details. Okay, so where do we start? Okay, I'm fond of starting off lectures by giving quizzes. Okay, and I've used this before. Some of you may have seen this. The question is really, how long has it been that humans have implanted synthetic materials into essentially their own bodies, right? So in this particular example, if you're a metallurgist, like some of my material science colleagues, I'll tell you this is from basically the Iron Age, so that's an iron tooth in the upper jaw or the maxilla of this particular patient. This skull was discovered in northern France, and you can pick which year basically this, or I should say how old this device has been in this individual. Basically, this is obviously an archaeological finding. And what you can see is in this X-ray, maybe it's a little light in this room, but the implant itself is tapered. So it has sort of a conical taper to the end. And largely, this is considered the first custom implant ever used in, at least discovered in a patient. And I'll just tell you today, because you're eating and having fun, that I used the SAT strategy, and I just picked answer B, okay, as the correct answer. So this is about 2,000 years old. So what's happened in 2,000 years with respect to materials we might use in the human body is something like this. This is from a cover of Science magazine roughly about 4 years ago. When I was in graduate school, there was a similar type of image from National Geographic that gives you the same impression. That is that we've taken all these synthetic materials developed from different industries, aerospace, textile, sort of clothing, manufacturing, and we've used them in a number of different implant technologies. I'll just point out a few. For example, the total joint replacements come from the aerospace industry. They're made out of titanium, aluminum, and vanadium alloys. They weren't designed to be implanted in the body de novo from first principles. All sorts of elastomers or polymers are introduced into the body. Of note might be things like the total joint replacement again, where there are polymeric components. There are vascular grafts that don't show up so well in this image. This is the total artificial heart, which uses basically polyurethane material, which if you're sitting, well, these are nice new chairs, but if you have a cushy, foamy chair, that foam is made out of polyurethane, right? So it's not really sophisticated, even in 2002, in terms of the level of materials that have been introduced into the body. So what happened probably somewhere crossing over with this idea and moving into this idea of this bionic human, where you can tell these are all more or less synthetic materials, and a lot of them, I guess related to CITRIS, have some sort of electronic control and potentially electronic interfaces for remote sensing and maybe manipulation of the device. But if you think of this device, this is a simple vascular device. It's called an intravascular stent. And it's deployed inside a blood vessel with a very slick strategy of using basically a balloon. So we call this a percutaneous delivery of this, and we call it minimally invasive delivery of a biomedical device. And the balloon expands this stent, this structure. You notice it's sort of a fenestrated, sort of hollow cylinder. And it's made with laser micromachining of stainless steel. So this is no different than your stainless steel cookware, right? It gets back to my original introduction, or part of this, which is, these materials are not elaborate in terms of what they were chosen to do as they interact with the biological system. If we look at one of these struts in cross-section, we see this is the initial tissue of the vascular structure. This is the cross-section of the stainless steel, and you can see there's new tissue forming over it. We call this a neointima, or essentially a new, basically, layer of tissue that's formed over this material. This is the basis of the concept of biocompatibility and how thick this material, basically, this new material or new biological material becomes as a function of interaction with the implant itself. So how does that happen? What drives this interaction? Well, it really depends very little, if you really want to think about it, in terms of the bulk physical properties of the material, other than that physical property is what makes this device work. In other words, it has to keep the blood vessel open after you basically eat too much fatty foods or rich in sort of essentially cholesterol and fatty acids and so forth. But the real interaction at the material side occurs at the cellular level. These are different cellular components. Proteins are going to be associated with these cells. And one idea we came up with somewhere in the early 90s was if you could take a solid implant material like this and functionalize and decorate its surface with some sort of biochemistry, that the cells might be confused and think that this synthetic material is really an extension of what they normally see. Right? So along the development of this idea, which is improving, I used the word already, I didn't maybe define for you, but I used the term biocompatibility, and that is to create a material system that is more compatible with biological tissue. And so I've sort of discussed these implants already. This is a total joint replacement. This is the intravascular stent. And the basis for these is really in the cornerstone of material science, and that is the material has some structure. You process that material and you alter its structure, which influences its properties, and ultimately you have a performance of that device. And that is sort of the universal paradigm in material science, biomaterial science, whatever you want to call it. What becomes more or less a difficult problem is things like after a heart attack, if you have an infarct in this area, you're not going to repair that with a stainless steel mesh. You're not going to repair that tissue with essentially classic materials, polymers, ceramics, and metals that we use and you might find around this room. Same thing if you have a congenital defect in the skull, which you might want to regenerate bone. You typically wouldn't necessarily call on the materials I just identified. And so this led to sort of this concept of tissue engineering. And the idea was that somehow in tissue engineering, you could redevelop or recapitulate the regenerative process and create something outside the body and then implant it. And so the Greek mythology analogy to this is Pavilion's dream, in a sense, where you have this sculptor who carved this beautiful statue out of ivory and then fell in love with the statue. And so as this sculptor falls in love with the statue, Aphrodite basically brings the statue to life so they can wed, be married. And so now you take this synthetic material and you breathe life into it. And this is sort of a mythological paradigm for the field of tissue engineering, right? So, but what allows you to go from this statue to this living one, right? So you have these components, and these components are the cornerstone and the structure of what would be considered the modern field of tissue engineering, biomaterials, and regenerative medicine. And they're all sort of folded into one because they're sort of interrelated by the nature of what might be the end goal. And the end goal, I'll just start off with that, is to regenerate various tissues. This is cartilage, this is bone, and this is a histological section of cardiac muscle.
Are they all the same magnification?
No, they're not.
Oh, okay.
Okay. But we'll get into this one today. These two I won't talk about today. But this is an image of human embryologic stem cells grown in our lab. And that's a colony of cells. And I'll describe how we actually achieve this goal. But the point of this slide is to just tell you that you need really three components. You need stem cells or some sort of cells that are capable of becoming other cells in the body, or we'll describe in a moment, more differentiated cell types. And you need some sort of matrix or some sort of system that these cells can grow on. And then you need instructions. And instructions for those cells typically come in the form of soluble biomolecules. And so these soluble biomolecules are growth factors. Sometimes we call these things morphogenetic proteins, can help generate this response. And there's two different sort of camps. And one is that this, and I'll describe one or two examples of this in a moment, this ex situ tissue equivalent. And the word equivalent is used loosely, but the idea is to generate tissue outside the body that has some equivalent capacity to your normal healthy tissue. So the idea here is maybe, and I'll show you an example of making an artificial blood vessel. And then there's another group that sits in the idea of what's called this in situ development inside the body, in vivo. And that's more or less like the concept of regenerative medicine. Like if you have a defect, regenerate that tissue in the site with the body providing the bioreactor and all of the capacity in the sense of fulfilling the need to regenerate that tissue. Okay, so...
Can I ask you a quick question?
Yeah.
I don't want to get into any politics.
No. But can I just say, if anybody has a question, stop me, because this is sort of a general audience, and so I may use terms that aren't really familiar with, or you might not be familiar with, so please ask for the true definition.
Without getting into any politics or anything like that, could you do anything like this without stem cells as a host cell? Or?
Yes. Matter of fact, but stem cell is the term is important to differentiate between adult and embryologic stem cells.
Oh, okay.
And the stem cells that we developed, this is actually ex situ with cells derived from cartilage that we obtained from the slaughterhouse when we were in Chicago. Okay. And if you want to hear more, when you're finished eating, I'll tell you some more details on that. Actually, I was going to tell everybody to hurry up because we've got a few slides that maybe you don't want to see. So chew quickly. Okay, so the artery equivalent, what they did, and this, Paul, this partially answers your question, is they've taken this we call them sort of polymer fleece or a synthetic material made out of polyester. But this particular polyester degrades into, by hydrolysis, into products that the body can handle, normal metabolites. So if you've ever had a synthetic suture, this is the kind of polymer that these materials are made of. And this gives you the scale. And the idea from this is from Laura Nicholson's group, who's now at Yale, but she was at Duke and MIT when she did this work. Seeding the cells in this structure, you can see it has some porosity to it. It allows the cells to sort of move in and attach. And what cells do when they sort of integrate with a material like this is they create their own, what we call extracellular matrix. So they synthesize their own repertoire of proteins that allows these cells to then let this cellular milieu or the cellular components that they generate take over this system. So this is all artificial, but over a period of time it will be completely replaced by biological components, native proteins. You know, if you grab yourself, you can feel your skin and so forth. Think of your arteries as having this sort of flexible, or what we would call viscoelastic nature in terms of its material science. So with that, you can see these cells. This group created a conduit of this material, seated cells on top. They were flowing liquid through, so this thing would pulse in this sort of bioreactor, and I'm not going to go into the details this afternoon, but by creating the appropriate mechanical forces on these cells as a function of also putting them in the right biological context, they were able to create something that was pretty close to a native artery. This is it in cross-section, and the strategy is that this material has blood would flow through. This would be circular. It just, the artery isn't strong enough on itself, so when you take the histological section, it kind of collapses down. But this was circular before this process. It has a layer of cells that would be consistent with the normal blood vessel because they introduced those cells. So they can almost make this arterial equivalent with using cells. In this case, they derive cells both from porcine or a pig and also from cow. And theoretically, you could do the same thing if you needed these cells. You can, just so everybody knows the real surgical problem, if you have an infarct, you usually take your saphenous vein, which is in your lower leg, and you basically put that vein on the arterial side, which is a totally, from a biomechanical standpoint, kind of a bizarre thing to do because veins are really elastic and flexible like a balloon, and arteries are much stiffer. Okay, and if it wasn't that way, when the heart would pump, your arteries would balloon out, and you would never have the pumping capacity in your heart to drive the blood throughout your whole body. So all the biofluid mechanics guys are shaking their head yes.
So can you use the strain and stress on these cells to change the tissue structure and
Oh yeah, that's a really good question that will totally derail what I have to say. But I'll get to that point maybe in about 10 slides. But that's a real hot topic right now, and actually an issue of Science earlier in March. In fact, it's the one with Luke Lee's work is on the cover. A colleague at Penn, Dennis Disher, I'll show you some of his slides in a moment. The whole idea is to introduce mechanics and chemistry into material, and the idea that the mechanical properties will influence the cell behavior and tissue development. And so we get back to this question in terms of what kind of cells are important here. It turns out you can do this with adult cells. It turns out the real details of the story is that these things do not have the same burst pressure or can't handle the same pressure as our native vessels. So there is some problem here in terms of what they produce is not exactly the same in terms of their biochemical, we'll call it again, biochemical repertoire. The details are important in terms of what those proteins are and how they're organized, just like in any material, in any structural material. So the adult stem cells work, but not enough where we would have this as a clinical device. And you notice this is a 99 paper. They were probably working on this over 10 years ago. So it's slow inroads. I'm just going to show you quickly a colleague, Vladimir Mirnov, extremely creative person, has these ideas that you can take things like basically printers and use sort of what we would call immediate sort of CAD-CAM design, plot out groupings of cells, right? And so, these were not individual cells, but masses of cells. And you could then let those essentially cells grow this up in any type of, obviously it's a printer, so you can create any pattern you want. So now we're not just talking about individual cells sort of seated and letting them go anywhere. We're talking about positioning those cells structurally in terms of a context in a 3D environment, right?
Do you mind going back to the
yeah.
So
it's a cartoon, but
has he built this or?
Yeah, I'll show you some data. They have built this. They've basically modified an Epson printer, and this is actually, I'm sorry if I didn't. I was just going to gloss over this, but this is this red part is media, meaning liquid that holds the biological cells, and then they're just spit out in the same way as ink droplets, but they have modified this so that it can handle the larger size of the aggregate as opposed to a droplet. I'm
pressing on this because we have these rapid prototyping machines in the lab. I mean, what's the kind of size of the droplet that comes out?
Right. So and I reviewed a lot of SBRs for NIH for years in this whole field of rapid prototyping, mostly to make these kinds of to make these structures. Okay. Right. So all sorts of stereolithography, all sorts of systems to make these 3D structures. I think has a role, but ultimately that's sort of setting the template to get to this next spot, which are next position.
So Dan, what's the droplet size of your printer?
It can vary.
Well,
So I mean from 1 micron to 2 millimeters.
1 micron to 2 millimeters.
So this
That's
range
how
would be, this would certainly be in that range you'd want. So typically a cell is 10 microns in diameter,
Okay.
maybe 20, and you might want to plot individual or aggregates of cells. So this would be in the range. I actually gave a lecture at Park last summer to talk about, you know, how they can use their printer head technology, and they have all sorts of ideas how that might be exploited. And we can free that up in the discussion. But
How do they print them into a flat surface and then it becomes a component, or?
well, in this case, I'll show you an example where they printed them in a sort of a z-axis. So they printed it, sort of made these Let me show you what they did. Is they just took cells and they just aggregated cells and they just stained the membrane orange and green. And so they you know, sort of wanted to show how they eventually will fuse over time. And these are the aggregates of cells. By the way, this is sitting in a material, a gel, called a hydrogel. And the hydrogel isn't imparting any information, but it is keeping the cells sort of in a collected environment, right? And so over time, you can see that here's the time scale. You can see that these fuse into nice little sort of, they would be ring structures. And now they've done the green, orange, and so forth. And so I did want to show this this is not our work, but I want to show this group because of what you're bringing up, the capacity in the Berkeley environment to create these kinds of things, which I think are quite exciting in terms of the next step in creating tissue structures. And then don't, if you're eating, stop. Okay, here it is. Okay, they can create something complex as an ear, right? So they did this kind of thing, and they put chondrocytes on it. So these are terminally differentiated cells that are of the cartilage lineage. And you can see you can create something like a human ear, right? And where would this be useful? Well, cosmesis and plastic surgery. So you would have, you know, the desire for somebody that might have lost an ear in an accident or nose or where you need cartilage built up, you could instantly have this available. Now, the details of this, of course, are, you know, would you have an immunologic reaction to this kind of system? If it's your own cells, you wouldn't. So if you harvested cells from your own body, we could grow them up in a bioreactor and then do this. That's a potential therapy. Okay, so what's the that is sort of the introduction to get us to today's lecture, which is really why then human embryologic stem cells? What role would they have in tissue engineering? Well, the first thing is that these cells have what we call a sort of a multipotent or totipotent potential, meaning they can derive from, and I'll describe where you get these stem cells from, but they can develop into all the different tissues in our body. And that's, I think, the important part, is that there's a theory, although nobody's proven this yet, that, you know, all our cells come from one source cell or a cell in what we would call a physiological state that can be told to go in any direction. It would be like all of us were in this room, we're all stem cells, and we're each given basically a roadmap to go somewhere in the country or the world. And we go there by taking different modes of transportation, which is the metaphor for the physiological, could be mechanical, and also basically chemical cues to get us to the right, basically, tissue. Right? So, you know, just say these are the embryologic stem cells. This is called the inner cell mass of a fertilized egg, which develops into something called a blastocyst. So this is a sort of spherical layer of cells, and then this inner portion develops into all these three tissues, and it's not important, but they have different what we call germ layers. So an endoderm, an ectoderm, and a mesoderm all are components that are derived from how this develops, and then a more complex structure. So in human embryologic stem cell work, this inner cell mass is harvested. And as this inner cell mass is harvested, you then grow these cells up in a tissue culture environment. So the tissue culture environment you grow these cells up in looks something like this. So this cell basically is harvested, or this group of cells is inner cell mass. So isolated inner cell mass. You notice they're sitting on something else. This is a layer of another cell type. It's basically a fibroblast, which is consistent with coming from muscle tissue, but it's also essentially an irradiated layer of cells, which means these cells are going to produce molecules into this culture system, but they're not going to grow, theoretically. So the idea is that these cells, these feeder cells, support these cells in their growth. And this is largely, this culture mechanism is largely derived from what was observed when trying to grow mouse embryologic stem cells And so this technology was borrowed from that field. There are significant problems, though, with this. And I'll get to those in a moment. But to take the idea then, if we have these cells, we ultimately want to replant these cells back into the body. And instead of just engineering a tissue equivalent, we may want to do things that are more or less radical or more or less challenging. And that is possibly using these cells, if properly conditioned, to target diseases of the brain like Parkinson's and so forth. Again, I'll give you the example of the ischemic heart, and then also bone regeneration, cartilage regeneration, with the theory that these younger cells are going to behave better in the environment than the older cells. The problem is that these cells are very difficult to grow up in culture. So they have uncontrolled behavior in cell culture. So this is not a solved problem. And I'll give you an example of how we're working to solve this problem. It turns out if you think about some tissues that use other cell types for regeneration, on the order of about 1 million to 10 million cells per ml is needed for different types of therapies. What that means is that you need trillions of cells to get this to work. So one of the first goals really is to get a process or develop a process where you can generate a large number of cells that can be used subsequently.
So is that because you need some activation energy to get it go, or many die in the process, or what's the
part of, I think it's just a lot of unknowns in terms of how these systems work, and that it's known you need these so-called cold culture of fibroblasts to feed or help contribute to the environment. They use the word microenvironment, meaning the community.
So they're actually working together as a kind of complex interconnection.
Exactly. And then there's the fact that because the signals aren't ideal, the growth kinetics of these stem cells is not optimized.
Yeah.
So I'll give you an example of a reductionist approach we've taken to try to alleviate that problem. If we implant these back into the body, they have poor survival. So in the cardiac example, if we implant those cells, almost 100% of those cells die off in 2 weeks. So any benefit of stem cell therapy is largely sort of mythical because these cells are dying off. So how can you prevent these cells from dying off? That's a big problem. So the push for Prop 71 was this part of it, right? Nobody sort of discussed this problem. But to push these cells back into the body for some therapeutic use, and this arrow may never develop, really, right, honestly. So there's a lot of hurdles even to get to that point of the process. So in our hands, the first thing we worked on was this so-called self-renewal process, which is to get these things to basically replicate in high enough numbers without actually differentiating. And so with that said, if anybody's really interested in this, go to this website where I took this part of the image. It's basically stemcells.nih.gov, which has a really nice tutorial, about 200 pages, to fill in a lot of the details that are obviously important for this kind of work. What I haven't said yet that's pretty important is that these feeder layers contribute contamination, I'll just put it that way, to this group of stem cells. So they're somewhat contaminated by a combination of, first it's xenographic, meaning 2 different sources of tissue for 2 different species, as well as you have oligosaccharides that are represented on the human cells that have the mouse we would call the mouse repertoire in terms of their chemistry. So somehow the human cell has been sort of perturbed by the mouse cells. And so the idea is to get rid of all of that in terms of the chemistry. So this goes back, somebody just asked me this, what happened with Prop 71? So I gave a lecture on Cal Day. So the morning of Cal Day, I opened the paper and my computer has a little camera here, right? So I took a picture of myself telling you that basically the judge has okayed the stem cell program. So that's pretty good. Gatam, all showered and shaved and everything. So with that said, then, the idea is back to, we might eventually see some of this Prop 71 money. But in the interim, we still have these same problems. How do we get this inner cell mass to grow? And what are the real technical challenges that we see that we need to look at? So one is this expansion in cell number without the cells differentiating or becoming what we would call a cell type that is consistent with a tissue. So we call that differentiation or maintenance of self-renewal. We ultimately, I didn't say this, but before you can plant these cells back into the body, they need to be differentiated, or you need to commit them to a certain cell lineage. Otherwise, they'll form a modified tumor, which we call a teratoma. Matter of fact, that's a check to make sure these are true embryologic stem cells. If you implant these, you will get a teratoma with all 3 germ layers that I mentioned. So that's actually an experimental protocol to verify their stemness, if you will. And then, ultimately, you got to get promotion of these cells in functional tissues. You have to integrate them into the tissue without them dying, right? So today I'm going to talk a little bit about each of these conceptually, but I think it's important to know that it's a part of not only just material science, but cell biology, automated instrumentation and imaging is important, and then systems biology would be an important component of this as well. And so you look at this Venn diagram, this little intersection here, and that's really where I think that kind of collective sort of collaboration, which probably falls in the domain of sort of CITRIS and a germane approach of trying to bring a number of different folks together to solve the problem. Okay, so what is this? So this is actually what a stem cell culture looks like. These are the mouse embryologic feeder cells. You can kind of see them down here. This is the border of the human stem cell culture. You can see the morphology of the cells is quite different. They have a much higher nucleus, which are these little black dots to the sort of white border. So we call that nuclear to cytoplasmic ratio. They can also be stained in certain ways to define whether they are embryologic stem cells. Here is, again, the colony. This particular class of stem cells is one derived from UCSF. So this is the human SF6 cell line. These are, again, we'll avoid the complete politics here, but these are what I call the Bush cell lines. Okay, so they're the ones the president defines that I can't get thrown in jail, or my students can't get thrown in jail with me if we decide to work on them using federally funded sources. We can work on these cells and many other cell types if we use state funds, and that's the whole idea behind this Prop 71. If you're not familiar with it, there are some other issues about isolation and really sort of almost legal-like interpretation of what one can do with federally approved sources. Okay, so this gets back to that early question about, when you seed these cells on this material, does substrate elasticity play a role? And I'll just tell you quickly that typical cell culture systems have a modulus, or basically a stiffness of a material, somewhere on 10 to the 8th. But our native tissues are about 5 orders of magnitude lower. So if you look at this sort of thing like collagen gels or collagen, which is a major protein in your body, you tend to see that they're far from basically these materials that one uses typically to culture cells, far from sort of our native tissues. So we've made a series of materials that look like this, also some that sort of span in this range. And the concept behind using those materials is that they match sort of the physical properties of functional tissue. And so the idea really is, can we create artificial materials now that are designed from a physical basis to integrate with biological systems, just on the mechanical side? And this is a newer concept. If you remember, all the materials I was showing you earlier are very hard, sort of high modulus materials, right? Nothing is really that soft and sort of compliant with the mechanical properties we would normally see with functional tissue. So, this is for Rich, right? So what do? Well, sometimes you use biology to inspire what you do in different parts of the world. So in this case, I was inspired by this sleeping, it's sleeping, see its eyes are closed, kangaroo. I had one of my friends take a picture, but it's to introduce this idea of biologically inspired. So use biology to inspire what you're thinking about. So in this case, we've also called this for years biomimetic materials. And, oops, went too far. The idea is that you use both the concept of mechanics, so complex modulus or stiffness of a material, and its biological content to affect some response. And so what you really want to do is sort of map response surfaces that define the idea that the biology in the system has both a mechanical and a chemical component, and then identify that aspect of the material that influences self-behavior, or behavior of cells. So in this case, self-renewal would be a short-term example to get those stem cells to renew without differentiating. But ultimately, we might want them to differentiate, and I would expect this system, we call this a response surface, to be totally different than if we were just looking at proliferation. Right? So the idea is to make materials that allow you to test this kind of process, and we call these sort of materials are tunable with biophysical properties, meaning both their mechanical and chemical signals. And I don't have a complete map for stem cells, but here's another cell type that we've used in the lab. This is actually an osteogenic cell, so an osteoblast, which produces bone. And this is just to convince you that you can get material behavior, or say cell behavior. In this case, this is cell growth, where you look at the stiffness of the material, and you can see some increase. You look at the concentration of, this is a biological ligand that's introducing information. So the RGD is letters for specific amino acids, so arginine, glycine, aspartic acid. And this is a function of concentration. What you see in this map is that we really haven't saturated it. As we increase the stiffness and increase the concentration, we get essentially better response. So with that, that's kind of where we're going with some of these stem cells. And so what we do is make a material that has a chemistry in itself, that the chemistry is defined in a way that the stiffness and the mechanical properties are defined by this matrix. And then we introduce, I'll tell you what that means in a moment, we introduce biological ligands independently. So this is called a semi-interpenetrating polymer network. And these ligands that are introduced independently have signals on them. So this part contains biology. This part contains essentially the mechanical properties. And as we can create these things independently, we can create those maps and those response surfaces and try to probe how a cell might behave under different conditions. I'll get to this later. This just means it's thermoresponsive, which is kind of fun, and I'll show you why that might be important. And the idea is you can introduce different ligands, different signals, and ultimately these signals are what the cells respond to. The cells respond and they come into this material, and now instead of the material just degrading, as I said earlier, by hydrolysis, I won't really go into this too much today, a little more biochemically related, but we can define these cross-links that hold this system together such that the cell secretes enzymes and degrades this. And I'll show you an example of something we don't really understand, but it's kind of neat that showing you that cells do that. So here's the chemistry. I won't really tell you much about it today, except we take these polymers, we polymerize them in the presence of a cross-linker that is biological in nature, and we basically create this network. And I showed you this cartoon. So the network defines the physical properties. The introduction of this linear chain defines the biological. And in that case, then you get a material system like this. So this is just, it's really the data on the left I want you to pay attention to, is that at room temperature it's kind of flowable and it can flow out of this petri dish. But at body temperature it's heated up and it goes through sort of a phase change that allows it to stiffen. And I just want to show you then we can measure those properties. And an example here, if we were relative to collagen, for example, you can see we can make materials with various stiffnesses as a function of how they might be loaded. So we call these environmentally responsive materials. And here's what we've been able to do with some of them. One is to take, now these are the same human embryologic stem cells, but you notice something's missing. This mouse feeder layer is missing. And the mouse feeder layer is missing for a number of reasons we think it's important. One, it contributes contamination to the culture. Two, if you're going to try to harvest these cells and then distribute them for therapeutic use, you don't want another cell type, especially from another species, contaminating this system. So you want to sort of separate that whole part, that mouse feeder layer part, out of the equation. And one of my students, Ying Li, has been able to do this. Has worked very hard to create the synthetic ECM, we call it, extracellular matrix, that allows for this to happen. So this cell colony does retain its stem cell behavior. And an example of showing how that's dependent on the material properties and not just basically any material will work is even in this system that we've created, if we change the concentration of this signal to the cell, this RGD signal, you can see that very few cells attach in this upper left image. The right, these cells on the border, these are called differentiating cells. They've come out of the colony and now they're starting to differentiate. So they don't have the stem cell, they don't retain their self-renewal process. This is a problem because now we have, if we use this system, we have a mixed culture of these differentiating cells along with the real stem cells we want. So if we're going to differentiate the cells, we want to choose at a certain time when we can do that. In other words, we want to be able to differentiate the cells under our conditions, and we want to know what the cell is going to become. In essence, if we use a higher concentration, this is the image that I just showed you, we get to the result that is obvious, that these cells retain their self-renewal process. Just the final image in this part of the lecture is that this is what the normal culture conditions look like. It's staining for this transcription factor, or this component of the cell that's a marker for self-renewal. You can see it's very good on the normal conditions. On our cell system, it's also very good. I'll tell you why it's a little bit diffuse. But somebody's always asked, well, what happens if I just put like collagen down onto the surface of the same material? And so Ying was able to do that, and you can see that these cells are all differentiated. So it's not just a combination of having collagen chemistry. It's a combination of modulus and collagen chemistry. We haven't figured out what the ideal modulus is, but we know this is the modulus of this material that we've chosen here. You can see these still have some OCT4 staining, but all of these cells have differentiated, has the same problems I just mentioned. Okay, so this is sort of kind of interesting. The reason we get that kind of diffuse staining is that because we made this system biodegradable by enzymatic means or by cell demand, the cells can grow down into it. And this is actually, this flat part is the top of the cell culture, and then they've grown down in it sort of in this hemispherical shape, which we don't fully understand. Ideas maybe the cells are trying to get more tension and trying to create, recapitulate sort of a developmental process. But we know this is the first observation of this sort, and we're kind of moving along on some projects, but also have this one in the back of our mind to try to figure out why that might happen. Okay, so I'm not going to tell you, because I don't want to go over too much, I'll just tell you we make other things, like thin polymer films, and these thin polymer films are useful for controlling where cells go and where they don't go. And that's important for now, maybe starting to create defined tissue structures in 2D and ultimately 3D, and for other things I'll mention in a moment. So this thin polymer network resists the absorption of proteins, and if you can resist the absorption of proteins, then you can control where cells go down on an interface. And so you can make arrays like this. This is on a 3x1 glass slide that we worked on a couple of years ago, and that now in this array are individual cells. And the idea is that these cells are in different states, basically in different mechanical context. And the idea is, if you put this in a spatially addressable format, could you go in and look at these cells individually as a function of exposure to different drugs? Or could you then take this analogy to the stem cell sort of stem cell theme and use these systems to create what we would call disease-specific types of tissue architectures in vitro that you can use for drug screening and drug testing. The reason this is important, where the stem cells come in here, is that probably the first use of these stem cells will not be in transplantation, as I discussed. It'll probably be in the area of using these cells in a therapeutic screen if you can get them to differentiate into disease-specific states, like a diabetic cell, a Parkinson's cell, and so forth. Right now, we don't usually use human cells for drug screening. The rest of these slides sort of depict how one might do these kinds of things. These are various patterning methods we've developed in collaboration with other folks. I won't go into the details, but just tell you that you can use an elastomeric stamp or stencil on top of that same thin polymer film. You can use an oxygen plasma to etch that away, so those that do microfab are familiar with these terms. And then you can let protein adsorb only in the islands of where you ultimately want cells to attach. This is a small cluster of cells, maybe like 15 to 20 cells in that cluster. And so you can then start to use these to make all sorts of shapes and structures. Wait till this pulls out for a moment. So all sorts of shapes and structures. So this part of the slide is more important. You can make linear arrangements. You can make so forth little triangles, cell aggregates, individual cells. And you get the idea that ultimately you want to create more complex tissue architectures, and possibly using 2D as a starting template for this and these stem cells would be quite important. And the last image really just uses a slightly different methodology to make a co-culture of cells. So now you have two different cell types under various conditions. I won't go through the processing and just tell you that you now have, in this case, one cell type on the inner ring, another cell type on the outer ring. And if we were interested in now co-cultures where we can be more precise in controlling these systems, then essentially things like drug screening would be important. And also cancer therapeutics. You know, cancer is largely considered now a disease that's specific to a certain tissue. So lung cancer would be different than breast cancer. So the same basically chemotherapeutic protocol should not work on both tissues. And if we had in vitro equivalents of these tissues to work out these experimental protocols, this would be quite valuable. Okay, so in the last few minutes, I'm just going to probably just skip over and just tell you that quickly that you can get these cells to differentiate into different cell types in collaboration with David Schaefer and Chris Saha in joint student between our labs. He's developed ways in which not using these human stem cells, but using stem cells derived from a rat that are somewhat committed because they are derived from the brain, but to get them to respond to different conditions where he can control whether they're under the self-renewal process or with different soluble fragments, he can control whether they develop into differentiated components of the neural lineage, so neurons or astrocytes. And he's done metrics of sophisticated quantitative analyses showing that he can control this behavior at the transcriptional level, meaning essentially at the gene transcriptional level. And I'll just sort of want to get to the end of this. This is also on a similar variant of a polymer system that we end up making. But I do want to get to this last part about thinking about, well, how would we use these kinds of materials? And with that, if we could generate a material that would help regenerate the infarcted heart, that has the capacity to support these stem cells, it might look something like this. So that when you have a heart attack, you basically have necrosis of the area if the heart attack is severe enough, meaning that black sort of circle there indicates cell death. So there's no oxygen to this region. What happens over time is that the system starts to expand, or say the infarct starts to expand because of what's believed is the high stress in the regions that border the infarct and the native tissue. So we call it the infarct border zone. What happens is you have cellular death and then maybe cellular apoptosis or increased cell death. And what you have is an increase in basically the size of the infarct due to maybe increased stresses in the wall between the infarcted region and the non-infarcted region. So this leads to expansion, and it basically is a cyclical process to ultimately the infarct is so large that the heart can't pump in a normal way, so it starts to enlarge itself to basically pump and get a greater volume of blood move through the system, and so that you end up needing a heart transplant after about 8 years or so if this infarct size is large enough. Okay, so this is one of my students, Sam Wall, is working on this project with Judith Guccione and Michael Mann and Mark Radcliffe at the VA and UCSF Medical School. And so when you have this infarct, the wall thins, so these regions become high stress regions. So the regions in the border zone become high, basically areas of high stress, again getting back to the idea that mechanics can affect cell behavior. And so what we've come up with is if you inject a gel into this border zone, you alleviate the stress in this border zone in such a way that you can prevent the progression of the congestive heart failure or the expansion of essentially the infarct. So in this case, we don't necessarily yet have to regenerate or repair all the tissue that's damaged. But what we want to do is at least keep it to and isolate it to a certain size and keep it from expanding. If we put certain drugs in this thing, so we just call this a hydrogel, it's blue, but if we have certain drugs in it, maybe it will actually turn red because that means we'd increase the blood supply to that region. And so we can now have a region where we've actually had some repair. And if we had stem cells, maybe we can completely regenerate that tissue in line with sort of developmental processes. So here's where this thermoresponsive polymer comes in handy, is that we can basically take cells, put them in this gel, inject it into the heart, And here's an example of the hydrogel injected. Here's it coming out. Here's one of the variants of the hydrogel coming out of the syringe. In this case, we're showing it being sort of dipped into a beaker that's held at a higher temperature. You notice it's sort of clear and globular when it went in. It will actually shrink a little and turn white, and as it does that, it basically has gone through this phase change and it's become much stiffer. And so we have a system, this is the same hydrogel that we use to grow the stem cells on. We have a system that what we're trying to develop is we can grow these stem cells on, and we can eventually use that same system to carry the cells into the heart for this kind of therapy. This is just to remind me to tell you that Sam has done some really nice work and it's just about to publish this paper. It turns out that if we model, and this is with Julius Guccione using finite element analyses, if we model the infarct in the heart and then use the blue regions as areas where we inject this hydrogel sort of around the border zone, and these are the volumes that he modeled in his injections, that we can change what's called this ejection fraction parameter on the ordinate as a function of basically a heart, in this case, heart pumping, consistent with what is observed with clinical trials of just injecting the cells alone. And some may ask, well, why is that important? Because it gets back to what I said earlier, is that most of the time when these cells are injected by themselves, they're going to die, right? And they die rapidly. But they do have a physical property. You're not injected saline. You're injecting 6, maybe 20 million cells per ml, which has actually the consistency of like a thin paste. So it does have physical properties consistent with the modulus of the material we've modeled here. And so we think in one capacity that just having the material itself present will help alleviate the stress in the border zone. But that's not going to go far enough. What we really want to do is introduce these stem cells into the whole process. And so to give you an idea what it looks like, I think most of you are done eating, so you can look at this. This is a nice little video. This is a mouse heart, just so for you guys to all notice. This is a very tricky surgery or implantation. So the heart's beating about 300 and 200 times a minute. You're going to see this, the white stuff that comes up is the injection, and then the thermo setting. There it goes right there. The gel is now thermoset right in that region. And think we're congratulating each other, hey, it worked. But the idea is now you can take that system and use it as a strategy to transplant cells. And as you do something like this, you then have the ability to look at things like tissue regeneration in this process and as a sort of an asset for carrying in cells and so forth. I think maybe I'll stop here to allow for... I have another example with bone. It's the same story, though. Just to maybe just focus this a bit, that we've done this so far. This example is without cells, right? So this is just the material alone. The material itself can have sufficient capacity to stimulate regeneration, right? But the addition of cells we think will be much more important. And maybe I'll start off by asking myself the first question, which was related to the one you asked, which is, well, why would you want to do this in the absence of well, I should say, why would you want to do this with stem cells, embryologic stem cells, compared to adult stem cells?
Yeah.
And the first question, first answer would be, well, to determine that there's no difference, you need the human stem cells and the embryologic stem cells as a basis for control. So you need them to do the experiment properly. Right. And then we can really decide what's next. But before, I'll open this up to questions, but I do want to thank, so I just want to get an acknowledgement up to my students and our funding for this. Should be somewhere around here.
These are big files.
This is a big, yeah, all these images, yes. That movie's like 100 meg alone. Okay, so had a lot of collaborators, a lot of funding from NIH. David Schaefer and all these guys are on the heart project and also on the stem cell project. Chris Saha did that work I just described with respect to the differentiation of neurons. Eugene Chung and Jacob as a whole team, Sam Wall, all working on hydrogels, and Ying Li did all the work related to the project on the embryologic stem cell development. So thank you very much for your time.
Very good. Well, like all good semesters, it should end with the best at last. That was a fantastic seminar. Thank you.
Thank you.
So I'm opening the floor to questions. There must be a number of questions that come to mind. Yes, Jane.
So is there any cells that can, so what kind of cells can use this kind of method, or what kind of other kind of cells cannot, is not compatible with this method?
So your last question, I guess, is trying to prove a negative, which is very hard. So in essence, so far we've designed these systems in a way because of their structure and also the ability to tune their properties, they theoretically should work with any cell type. Where we can't say that positively is we haven't worked with every cell type, and neither do we ever plan on it. But with the cell types we've looked at, these systems work fine.
As long as you adjust the modulus,
We can adjust the modulus and the biochemistry, which I didn't even get into the real details of that, because we can introduce growth factors, we can conjugate them to the matrix, we can change, theoretically, we can change the kinetics in which they're released from the matrix. So all of these are very important processes from the cell behavior standpoint.
So I just realized we went quite late, so forgive people for...
I didn't have a clock, so I had no idea how much time.
Oh, that's okay. A couple more questions, and then we'll... Yes, please.
You said that just the injection of the polymer would trigger regeneration. What do you think explains that happening?
Two things. One, the signals we put in are the same signals the cell would use from extracellular matrix proteins. So it's not too unlike what they're doing. Plus, the crosslinks are the same crosslinks they would see in those proteins, or say the same chemistry they would see that they would have to degrade to move through the protein matrix. So again, this biologically inspired, biomimetic, we're stealing little pieces of the system. We have much greater control than just using a synthetic, native protein. And we can do a couple of things. Design better experiments, sort of for parametric analysis of how each component influences cell behavior. But we can also then introduce, as I said, these growth factors. And the growth factors, then you get sort of a order of magnitude jump in the change of the behavior. So this last image didn't have growth factors, but some of the bone work that we have introduced growth factors. We get significant amount of bone formation under those conditions.
Sir,
that phase change, is that reversible or irreversible?
Reversible.
And then rearranged?
It's reversible. If I didn't say it, it's possibly one way. Well, we have a problem when we culture these cells. If they come out and the gel's cool too long, the cells will pop off. This is a methodology developed by a group in Japan, and they can actually pop off sheets of cells using this anisopropyl acrylamide as the polymer. And it's called the correct term is a lower critical solution temperature. And what happens is the polymer is coming out of solution. So it's really like a negative entropy of mixing. So you have polymer-polymer interaction favored over polymer-water interaction. So this cell sheet peels up, or individual cells peel up, and then you can harvest them without using typically you use an enzyme which could damage the cell. So there's some advantages, but also some problems with using the same material.
one more closing question, and then... no, if not, we'll just thank you.
Thank you.